Describe the factors contributing to the molecular shape of proteins. So proteins are going to be polymers of amino acids. So to understand the structure, it's necessary to first understand the structure of an individual amino acid. What we're going to have is going to be a central carbon. Surrounding this central carbon is going to be an amino group, so an NH2 group over here. And at the other end, we're going to have a carboxyl group, so a C double bond to O and a hydroxyl group over here. Now, in addition to the amino group and the carboxyl group, there is going to be a hydrogen filling up one of the remaining bonds, covalent bonds in the carbon. And then the final is going to be this R group, this residue group, and this is going to be what is unique from one amino acid to the next. And so all of our amino acids are going to have that central carbon, an amino group, our carboxyl group, and that hydrogen. And then this R group is going to be variable and determine the characteristics of that individual amino acid. Now, the characteristics of those R groups is going to vary based on the composition of the side group. And so depending upon what functional groups or molecules are attached at that R site, we may have nonpolar amino acids. With these nonpolar amino acids, we're going to have a lot of nonpolar bonds, and so these regions will tend to contribute to a hydrophobic characteristic associated with the protein in this region if there are a lot of these nonpolar amino acids. In contrast, we've got a series of polar amino acids, and so these side groups, these R groups, are going to have distinct charge distribution associated with them, so differing amounts of electronegativity in their bonds, resulting in partial positives and partial negatives across this structure. In addition to that, we can also have amino acids that have a full charge difference, and so a shift of electrons, much like what we see in the formation of ions. And so we can have some amino acids that are going to have a positive charge and some amino acids that have a negative charge. And again, the composition of a protein is going to be determined by the characteristics of these side groups, of these R groups, contributing to the function of that region of the protein. So starting out, taking a look at the protein structure, the primary structure is simply going to be the amino acid sequence of that protein. And so amino acid 1, amino acid 2, amino acid 3. And they're going to be put together with a peptide bond. And so if we take a look at this slide, we've got, again, an amino acid over here, a second amino acid over here, amino group and carboxyl group. The side group in this case, we're just going with a simple hydrogen for that R group in both of these examples, mainly to keep the diagram clean when we take a look at it without making it too busy. What we're going to see is another dehydration reaction, and so what we're going to do is we're going to remove the OH from the carboxyl group, we're going to remove an H from the amino group, remove the water, and what's going to be left behind is going to be the formation of this peptide bond. Now, whether or not we have a dipeptide like we got here, so two amino acids hooked together, or an amino acid chain, so a polypeptide that is tens or hundreds of amino acids long, we're always going to have an amino group at one end and a carboxyl group at that other end. And so the primary structure is simply going to be the sequence of amino acids going amino acid 1, amino acid 2, amino acid 3. The secondary structure of an amino acid is going to start to have interactions with the carboxyl groups and the amino groups of amino acids, but not within the neighboring amino acids, but amino acids a few away on this polypeptide chain. And so what we're seeing is the formation of hydrogen bonds. Again, each hydrogen bond is going to be relatively weak, but the combination and the accumulation of these hydrogen bonds are going to give structural stability to the secondary structure of these polypeptides. And we could see kind of an organization of the protein in what is referred to as a beta-pleated sheet, or we can see kind of a coiled appearance or coiled structure to the polypeptide. And this is going to be contributing to an alpha helix. And both the beta-pleated sheet and the alpha helix are going to be held together by these hydrogen bonds between the amino groups and the carboxyl groups of amino acids within that single polypeptide chain. The tertiary structure of amino acids is going to further stabilize the structure of our polypeptide chain. Now, the tertiary structure we're going to see is going to be caused by or result from interactions of these side chains. And so if we've got hydrophobic R groups, so hydrophobic amino acids, they're going to tend to interact with one another to avoid interacting with water. And so a hydrophobic region of the molecule is going to be held together because of the hydrophobic characteristics of the amino acids in this region. We can also see hydrogen bond formation. Again, in this case, it's hydrogen bonds that are forming between different R groups, the side chains of these amino acids. Another example would be the disulfide bond. This disulfide bridge that we're seeing here, it's also referred to as a disulfide bond, where we're forming a covalent bond within the S side group, the sulfur side group of these amino acids, forming, again, greater structural stability to that region of the molecule. And then finally, the last example looking at interactions of these side chains would be an ionic bond. And so we would have a positively charged residue group, a positively charged amino acid, being attracted to a negative charged side group, a negatively charged amino acid, forming, again, some structural stability to the tertiary structure of this polypeptide. Finally, the quaternary structure, the fourth level of structure for the proteins, is going to be interactions of two or more polypeptide chains. And the classic examples to describe this are hemoglobin over here. So in the hemoglobin molecule, we're going to have two alpha chains, two beta chains that are going to come together to form the functional hemoglobin protein, which by combining with heme groups in the iron are going to be involved with the transport of oxygen throughout the body within our red blood cells. Another good example of a quaternary structure is going to be a collagen molecule. And so a kind of fibrous molecule that is going to be found within the extracellular matrix, the connective tissue, in which case we are going to have three polypeptide chains that are going to be twisted together. And the fact that these three polypeptide chains are twisted together, this is going to make this collagen molecule much stronger than any individual polypeptide chain. We're basically ending up with almost like a braided rope when we take a look at it in terms of its structural integrity and its strength at resisting stretch.